This is the second lecture for Friday, April 10th. And what we're doing is looking at this aspect of that's related a lot to oceanic ecosystems, but not exclusively to oceanic ecosystems. In fact, it has a lot to do with coastal ecosystems and organisms that are found there as well. It might be a life stage where they're out in the open ocean. But let's look at some of the different choices that you have. It's not like they're sitting there. Let's see. What do I want to do? What kind of dispersal strategy am I going to formulate? No, this is how they've evolved. The natural selection has led them to have these different patterns. It's probably a better term than strategies. But these are some things you can take into consideration. How much time are you going to spend in the water column? What is the timing of your spawning going to include? Not just the organisms themselves have to figure out the calendar, to have spawning take place synchronously when everybody knows that this is the time when you're supposed to spawn. But there's other things that have to do with environmental characteristics related to spawning. So there's a lot of organisms that spawn around the moon, the full moon, before the full moon, just after the full moon, something like that. Well, one thing is that the moon is their clock. The moon is the watch that organisms have. They don't have other ways of telling time. They can tell time with water temperature, day length, and things like that. But the tide, the moon, is a lunar phase, this 28-day cycle. That's a mechanism for telling time. Plus, you hopefully remember way back at the beginning of the semester that the moon is tied in with the tidal currents. And so it's not just a clock, but it's also indicative of potentially of water movements, water currents, high tides, maximum distribution, maximum dispersal. So timing is important. And then the second thing, that was a lot of information about the tides, but the time. Eggs. You have a choice basically of the size of your eggs and the number of your eggs. You can't have millions and millions of very big eggs. You can have a lot of little eggs, or you can have a few big eggs. Now that's going to influence what kind of larva you have, how long the larva is going to be able to remain in the water column. So a mother has got to decide, and again, this isn't she's sitting there saying, okay, I'm going to have 12 eggs and they're going to be this big. This is what has been determined by natural selection over thousands, millions of years. That this is the reproductive pattern that this species, this type of organism, has developed over time. But that's basically your choice. Do you have a lot of small eggs, or do you have a few big eggs, or somewhere in between? So these larvae that are released from the parents, there's some parental care to a certain degree. I mean, not for very long, but we saw that there's some marine invertebrates and some fish where the fertilized eggs that are developing aren't just broadcast spawned out into the water column, but there's some kind of care, or they're attached someplace. So they're not going to have very many eggs, and they're not going to be very big. You've got this type of larvae that only rely on their yolk. That's all they're ever going to get from the parents. Now those tend to be medium size and a medium number of those. And then you've got these planktotrophic larvae. Trophic refers to feeding. Those are larvae that can feed while they're plankton. Now if you can do that, you can have a lot of little eggs. You can sure, a lot of them are going to die. Most of them are going to die. You're not investing very much energy into any specific egg, any individual egg. you're producing a lot of eggs, and the strategy is that a couple of those eggs are going to survive. So we can have more little eggs because we're not giving them that much energy. However, they can stay in the water column. They can exhibit these pretty impressive dispersals by feeding as plankton. And if you're some little tiny organism that's floating around as plankton in the water column, then one of the things you can probably imagine is the longer that you do that, the more vulnerable you are to being eaten. You're some little tiny thing, some little speck of food, a little snack for all kinds of different organisms, then you're vulnerable. There's very high mortality. So all of these different organisms, though, they're floating around in the water column, and they're looking for some place to settle. If you're talking about invertebrates, a sea urchin, what are they going to do up in the water column? They want to be on the bottom. A starfish, a clam, a lobster, crab, they want to be on the bottom for the most part. So they need to find a place to settle. And it should be suitable habitat for them to settle. Well, what do you know as a little larva? You don't know very much. So there's different cues for determining where you're going to settle. So we'll look at some of those. But this is basically what we're talking about. You've got some benthic marine invertebrate. They spawn. An egg is fertilized by a sperm. You can look at this diagram and you can see this. You've got a fertilized egg that starts developing into a larva. And then if you're looking at this crustacean larva, then there's the chocophore larva. Then you see them. This is a mollusk larva. You can see the planktonic phase. They're going through these different phases. You get to the veliger larva, and now they're ready to settle. So they're going to settle down at the bottom, and then they're going to turn into some adult. They're going to go through these juvenile stages on the bottom as sedentary benthic marine invertebrates, and then they're going to reproduce. So this is the life cycle. The amount of time that they spend as planktonic larvae varies quite a bit between the different species, different types of animals. It's usually not very long compared to their whole life, but there are different lengths of time depending on the type of larva they have, depending on how far they're dispersing, depending on what their strategy is. But there's developmental stages that these go through. So they're growing and they get to a certain point. They're still not very big, but they're ready to settle. So that's an important thing. If you're an urchin, if you're a clam, if you're a lobster, if you're a barnacle, all these different marine invertebrates, most of them are benthic, so they're on the bottom. That's their way of life. So they've got to find a suitable habitat to come out of the water column, go down to the bottom, and then start your life as a benthic sedentary marine invertebrate. So how do you know where to settle? They're going to go through these drastic changes. They're going through developmental stages as planktonic larvae, and then when they get to the bottom, or they get to the point where they're going to settle, come out of the water column, they're going through even more developmental stages. So how do you decide where to settle? Well, there's been a lot of research into this. There's different ideas about different experiments that have been done based on the ability of larvae to detect these different aspects of the environment. And so the thought is that for some of them, there's physical cues that are really important. For some of them, there's chemical cues that are very important. For most of them, it's probably a combination of the two. But what do we mean by these two? Physical cues. There's a certain amount of light. You can move towards the light. You can stay up at the surface, orienting towards the light. And then when you reach these certain conditions, you know where to go, which is down towards the bottom, away from the light. So light is certainly it's a part of the directions on where to go. There's a lot of mechanical cues. You can detect the presence of bacteria. There's different sand grains of different sizes that tell you. There's different substrate types. So they could be going down towards the bottom, especially if they're in water that's not that deep. They can sense what kind of substrate there is. They have an innate sense of what substrate is suitable. They can also, supposedly, some organisms can hear waves crashing on the beach. They can orient towards those. And it depends on what their lifestyle is. If they're going to be attached to the rocks, of course, they look for rocky substrate. If they're going to be burrowing in the soft sediment, of course, that's the type of substrate that they're going to look for. There's also a lot of speculation that these larvae have fairly well-developed sensory systems that they can use to detect chemical cues. They can smell things, basically. Smell and taste things. So what is it that you can sense that gives you an idea, gives you information about whether someplace is good to settle? You're moving through the water, you're floating around as plankton, and you going through these developmental stages. You get to the point where, okay, I'm big enough, I'm running out of food, it's time for me to settle. I've reached this developmental stage where I'm going to settle. Now, usually after a certain period of time, you don't have a whole lot of choice. But you can wait. You can go at early in the morning. You can go later in the day. You're moving to different habitats. So you are moving around. You are experiencing an array of potential places where you can go down to the bottom. It's not like you're stuck in one place. You're being swept along. So you're experiencing different habitats, different substrates, and that's what you can look at. So I mentioned before some of the currents and the spawning methods. The larval dispersal is so that you can go out and you can increase the dispersal that you have. You know, it's not like you're just right next to your parents. So you're out in the water column, you're being swept along. Maybe the extent of that dispersal is a couple hundred miles. But you're swept back towards this suitable habitat, rocky shore, sandy beach, muddy substrate, whatever it is that's a suitable habitat for you as a plankton. These currents bring you back there. And then you can smell the adults. They put out chemical cues, amino acids that these larvae can recognize and orient to. Sometimes it's an attractive thing. Sometimes it's a repellent. So it depends on the life history of these organisms, whether they go towards the adults. Of course, the thinking is they're not doing much thinking, but the thinking is basically, if there's a bunch of adults there on the bottom, it must be a good place to live. That's where I'm going. Or there's a bunch of adults there. It's going to be crowded, so I'm not going to go right there. I'm going to go a little farther away. Sometimes they can detect other species that they would be attracted to, similar type of substrate that they're attracted to. But in terms of repellent that would inhibit them, they're smelling things, maybe predators, maybe competitors, maybe adults of their own species. And so there's a lot of interest in this. People have been looking at this for years in terms of being able to coat the bottom of a boat or pipes, something that would detract from all of these settling organisms. It would keep them from settling. There'd be some kind of repellent. You could put this substance that they smell and they say, no, I'm not going to settle there, rather than just having to scrape them off of the bottom of the boat and submarines and dive down and clean out pipes, as people have to do to a huge amount. Well, let's look at a couple of other things, and then we'll be done with this dispersal. We've looked at a whole bunch of different marine invertebrates, barnacles, starfish, mollusks. There's other things, crabs, jellyfish, cnidarians. There's other things that disperse. You know, there's a lot of fish that we looked at There's plants that are involved in dispersal. There's algae. There's all these different invertebrates that aren't stuck to the rocks that can move around to a certain degree. Crabs, lobsters, those kind of things. There's also all these cnidarians that aren't necessarily stuck to the bottom. They're floating around in the water column. So how do these things disperse? Well, for plants, pretty straightforward. These marine plants, one of the challenges for marine plants and one of the reasons why there's so few that survive and do well in the ocean is this difficulty of reproducing in salt water, having propagules, or seeds, in other words, that can tolerate salt water. They can last in salt water for a while and float around. We looked at mangroves and we looked at those propagules. So marine plants that can do that, eel grass, turtle grass, those salt marsh grasses, and mangroves, and other plants like coconuts, palm trees, and some other ones that are close to shore. They have some mechanism for being able to move around, either in the wind, water, tolerating seawater. For algae, they have these propagules. We saw this alternation of generation. They can tolerate seawater pretty well. For things that move around like crab, squid, some cnidarians as well, then it's not such a big deal. You know, these sessile invertebrates, which means they're stuck to the rocks, when they settle, they're making a choice that's going to last them the rest of their life. So when they decide to attach to the rocks, then that's pretty important, their choice. For these things that are climbing around on the rocks, they can move different distances. So the settling isn't as critical of a decision. They don't go through as many developmental stages. They might not rely on as many environmental and chemical cues to decide where to settle. And then in terms of fishes, they go through these different stages, a lot of these fish, if you had the planktonic fish, you could tell what kind of fish it was. You might not be able to tell what species it was, but you've got these little billfish that look like little swordfish, little sailfish, little marlin. And if they can feed in the water column, then they can last in the water column for a long time. Hundreds of days, which would take them hundreds, if not thousands of miles. Very wide-scale dispersal from these fish that can feed as plankton. And in terms of studying dispersal, there's a lot of reasons why people are interested. The populations, recruitment, why one year is a good year, another year is a bad year, what's happening with the population of lobster or crabs or fish, especially commercially important species. But it's not so easy to study as big animals, as you can imagine, or big plants. So a lot of what's done are these trawls, surveyed or planktonic trawls, plankton tows, where they're dragging things through the water. So here you see off of California, there's all these different sampling sites. Out in the Atlantic, there's all these different sampling sites for lobster larvae. The Gulf of Maine and basically between out on George's Bank, quite a ways away, where there's sampling that can be done. So basically, you sample different locations, looking for these different larvae, seeing what kind of patterns there are temporally and spatially, and look at the different characteristics of these different larvae in terms of how it relates to what the population of the adults are, especially the species that you're interested in, the fish or the invertebrates you're really interested in, especially from the viewpoint of harvesting them. And then there's genetics. You can identify them to species through genetics. And that's what's done a lot of times. These larvae it's hard to tell what species they are, but certainly you can tell by genetics. So that is for dispersal and last part of migration. That's a relatively long lecture, almost 50 minutes, that's longer than these lectures have tended to be, more 40-something minutes, the low 40s. So this will just have to be a little longer lecture for this Friday. But that is the end of migration dispersal.